Okay, cool. So I'm Eduardo, and I'm in Paul Sternberg's lab. And today I'm going to talk about single cell tools for WormBase that we have been developing. And for those of you who don't know about WormBase, WormBase is a model organism for C. elegans, which is this little worm that is like 1 millimeter long. It's transparent, it has 302 neurons, and people have been using it as a model organism for like 50 years. It's a really, really cool model organism. It always develops the same way, and it has a super nice community around it. People publish papers and then curate the information that comes out in the papers. And WormBase is this hub for this curated information. And so it's shepherded by Paul Sternberg, who oversees curators who are like biologists that have the knowledge to read the papers and input the findings, but also programmers who build the front end, the back end, the tools, and then members of the community who give ideas on where to go. And so today, I'm going to be talking about some work that was done by me, by Valerio, who is a software developer at WormBase, and by Paul, who is the PI. And I just want to start by acknowledging that we got a lot of input from the other people at WormBase, from the Alliance of Genome Resources, which is a consortium of other model organism databases that the NIH is trying to get to, come together and put a cohesive backbone for these things, and the worm community, especially the CeNGEN project, which has been spearheading production of single cell data with C. elegans. And so I always start my talks with this picture. Why do people want to work on single cell RNA sequencing? And the simple answer, the fun answer, is these two pictures. It's the difference between drinking the smoothie and eating the individual berries. When you drink the smoothie, you're averaging everything out. And if you're really interested in the different cell types, there are certain kinds of samples that you can't easily separate them. You can't easily tell apart what's going on, for example, in the brain. If you're working on something like the liver, then that's less of a problem. So because single cell enables people to dissect this heterogeneity, what is really different cell to cell in a tissue, it has exploded in popularity in past years. And so this is a figure that was published in 2017 on this famous article by Valentine, who was at Caltech in Lear's lab and is now in Boston. And he did this really nice review of where things were, the many different technologies, and how like every other paper was just like smashing through the roof, in terms of numbers of cells that were being done and numbers of the different clusters and cell types that were being found. And so this was in 2017, and this picture really drives home the point that the single cell was exploding. And then we did an update, and the explosion has continued, and it's not slowing down. It keeps accelerating. Like there's more and more papers are using single cell now, not so much for like method development, but really as a technique to do about fundamental biology, to figure out fundamental biology. And right now that is driven for the most part because of one particular technology, which is this droplet-based methods that are commercialized by this company, 10x Genomics, that makes it really, really easy to do this kinds of experiments. You press a button and you have your sample. Like It really is as easy as that. Put your samples in the right wells, press the button, wait 8 minutes. There you have your thing, follow the steps, put it on the sequencer, then you have your data. And so if you look at the breakdown, and this is a year old, most studies, the vast majority are using 10x, and the overwhelming majority are looking at human and mouse. And the other model organisms are like trailing behind, which to me feels a little bit like putting the cart ahead of the horses. But it's okay. The horses are catching up very quickly, as we're going to see. And I highlight C. elegans, which only has 5 single cell papers published so far. So after you get your data back from the sequencer, it's at least a FASTQ file that you need to align. And the alignment means 2 things. Not only you're aligning to see from which gene each read come from, you're also aligning to see from which cell each read came from. That's why you're able to tell apart different cells. And this happens because in the encapsulation here, each bead has a barcode represented by different colors. And so you can tell apart where each mRNA molecule came from. And so when you do that demultiplexing, instead of ending up with a vector that's just like genes per sample, you end up with a matrix that's genes per cells in the sample. And so this real alignment, it's done through either real aligners like CellRanger, like STAR, or CellRanger, which uses STAR, or through Callisto or Salmon, which do pseudo alignment, which is they just do perfect string matching. They don't do any kind of error correction. If there's a mismatch, they don't count. This gives you this big matrix that has every barcode that was seen at least once in the sample, and then you filter it. And usually, after this filtering, you're left with about 10,000 genes and 20,000 cells. And so the fundamental object in single cell data is this big sparse gene count matrix. It usually has 5 to 30,000 reads per cell, which is to say how many times you sampled a molecule, a PCR copy of a molecule in a cell. It usually has 500 to 5,000 unique molecules that were seen in a cell. And usually you see between 10 to 20,000 unique genes and about 10,000 cells in a run. And so what this means is that this matrix is like very, very sparse. It's mostly zeros. And a typical paper these days will have a few of these experiments. And so this is a bit of a new data modality. And how to deal with it, like the workflows are still very much up in the air. But there is one fundamental workflow that is you got to stratify your data, you got to separate it in groups of interest, and then you got to compare those groups of interest. And so stratification usually means you want to label the cell types that you have in your sample. Usually people do that through clustering, but you can also do that using marker genes. If you know, for example, that you have a blood sample and you know really which cells express which genes. And then once you have your groups, you want to compare them. You want to see what is different between them, which genes are going up or down, and then dig deep in the biology, because this part, this part is the computational workflow, but then digging into the biology is really where the really interesting stuff is. And so single cell has been putting out a lot of data, and because people are not really totally sure how to deal with all this data, kind of every other paper that comes out comes up with their own workflow because things are not settled. And it's good And it also causes a lot of confusion because it's really hard to navigate all the tools that are out there. There's this website, Single Cell RNA Tools, that tracks the data that the tools that are being put out with papers or just as GitHub repos. And if you do the math, every three papers get 2 tools. And so this has a few problems or issues that it causes. I think that the main ones is that these tools are often not super well developed because they're done like ad hoc to solve one specific problem. And after publication, they are not often maintained. And the other thing that happens is it's hard to discover, and it's hard to compare tools and know what should you use, what makes sense to use. A lot of them are not super principled, and so at this point, I think it's clear to everybody that some consolidation is desirable in terms of the methods that you use for single cell. And the question is like, when can we build on the work of others, and when do we really need to make a new tool from scratch? And so the tool that I use for processing all of my single cell data, and I'm talking just about going from the gene count matrix to doing whatever you do with the gene count matrix, is called single cell variational inference, or SCVI. And it's a PyTorch based framework for all kinds of single cell omics data. It has been validated and provides scalable models for single cell RNA-seq, but also for other modalities that sometimes include proteomics. And the really powerful thing is that it enables you to add a new model without having to do everything from scratch, and without to put out a separate tool that usually you're going to be the only one looking at it. And what is really nice about SCVI is that it has been around for a lot of time. In single cell time, 3 years is a lot of time. And a lot of people have contributed to SCVI, which is more than you can say for the vast majority of projects. If you go to the Single Cell RNA-seq Tools website, most of the projects are going to be 1, maybe 2 people working on the code base. And SCVI has like a lot of people that put serious work, which means that not only it's going to keep being developed going forward, but also that the current code has many eyes looking on it. And so, fundamental problems, errors, and bugs, which do happen very often in scientific software, are less likely. So I'm just going to spend 2 more slides talking about SCVI because it's a little different than what people usually do for single cell analysis. So there is this new preprint that came out in April where I am an author. So full disclosure, I wrote some documentation for SCVI and they invited me to be here. These are the models that are currently implemented and the tasks that they currently do. So there's a couple for single cell RNA-seq data because that's the most popular stuff. There's one for ATAC-seq, for site-seq, which is protein. And then they have spatial models, which are pretty interesting. And then Scartris is for integrating and doing transfer learning across models. And basically, the idea with single cell is to, with single cell variation inference, is that you have these modules that have that you develop, and then you add, you can use pre-coded tools like data loaders and training managers and all the stuff from PyTorch, but already packaged already packaged to deal with single cell data. And so your focus when you go and develop something really is on defining a model that makes sense, integrating the domain knowledge that you have into a machine learning model that makes sense to make something that is scalable. Scalable in the sense that like can deal with a lot of data, because that's what we need right now. And so just so you have a sense, like this is what it usually means when I say like a model. SCVI makes it very easy for you to create generative models, which are a different way of processing and analyzing data than people are used to. Because what a generative model does is you learn what is the distribution that generated that data like. And it's like if you have a coin and it has a certain bias, and you just sampled how many coins, how many outcomes you had for a given number of tosses, now you can parameterize a coin tosser generator. It's kind of like the same thing. This SCVI is a data generator. It learns how the input data look like for each gene and for each cell. And then it can generate new data that looks like that input data. And what you can do with a generative model is that you can compute any statistics that you want. Usually people just want to know, like, is my gene more or less expressed? But any kind of statistic that you want to compute, you can do with a generative model. And so this is what the single cell tools for WormBase are using under the hood. You don't need to be familiar with this to use SCVI, especially if you're just using the high-level tools like differential expression. But if you want to come in and start developing like models for your own use case, let's say that you have like temporal data, or you have like specific kinds of structure in your experiments, you know, not just experimental and control, but there's some other kinds of hierarchy in your data. SCVI really offers an interesting platform on which to do model development if you have a little bit of a machine learning background. Okay, so that's it for method development. I want to take a step back and consider where the field is going to go from now, that like the dust is settling and we're starting to like get out of the weeds of method development, and we're starting to try to ask, like, what are we going to do with all this data, what it's actually good for? You know, I'm tired of doing like all this technical development. Can I do some interesting biology? And C. elegans is a particularly interesting case for single cell, because all the cell types in C. elegans are super well characterized. One thing that happens is that in single cell is that the more you sequence, the more clusters you have. And so people keep finding new cell types. Like people keep finding more cell types in the brain, for example. With C. elegans, you only have about 1,000 cells. You have 300 neurons. Once you characterize them, you're done. That's it. And in certain kinds of tissues, in like human and mice, for example, blood, this is already the case. It's straightforward to annotate your data. This is happening. This is coming very soon for C. elegans, and a little later it will also come for other organisms. And the reason why this is cool is because now you don't have to worry about annotating your data endlessly. It enables you to focus on the biology and do cool things like, you know, multiplexed perturbation experiments without spending weeks on end annotating your data. And to drive this point home, like this is really, really important. Annotation is a huge pain in the butt that nobody likes, but everybody has to do. Here's a testimony of a friend that has been working with a brain data set, a new brain data set. So they have like more of glial cells than any other reference data set out there. He's been spending months annotating and re-annotating the data because it's hard, because there's not a lot of consensus on what defines a cell type. And it's a challenge for things like the brain. And for C. elegans, it's like there's a lot of consensus already on what to call things. There's a super well-developed gene ontology. So all these huge challenges for other organisms are very surmountable with C. elegans, and very quickly. And C. elegans is really getting there quick. So this picture shows the life stages of C. elegans. It starts as an embryo, and then it goes to this L1 larvae, L2 larvae, L3 or L4 adult, and then dies. And it can go into this like dower stage, where it's like in hibernation if there's not enough food or the environment is harsh. And I put a green check mark for the life stages for which I consider that the data that's out there is like really good. And so about half of the biological states, fundamental biological states of C. elegans have been surveyed with single cell. And I'm not saying it's super straightforward to do all the rest, but the task is laid out ahead of the field. And now that there is reference data, it becomes easier to annotate the next bunch. And once you have a reference for everybody, you can stop focusing on this technicalities, building these atlases, and then really go for the biology. And so what this means is that pretty soon we are going to be over this like hump of technical development that's really not that biology driven. And usually these days what happens is that, there's a big divide between the dry lab and the wet lab, and the biologist asks something from the bioinformatician who does differential expression or whatever, and comes back with a spreadsheet of results. And then the biologist looks at that and says, oh yeah, but not quite. And once it's straightforward to do the labeling, and all that you want to do is really this group comparisons, we need to have tools that enable this kind of visual data analysis without intermediaries, without you having to wait a month or a week or a day for someone to understand what you're asking and come back with the results. And we have the pieces that we need to build these things. We're starting to understand what is the right or better ways of doing single cell analysis. And so we need to have better interfaces for doing this analysis. And before I go further, I just want to highlight that, like, when I say visual data analysis, I don't mean like looking at t-SNEs and UMAPs, because this is how I feel when a paper figure 1 is showing me the t-SNE. It's like it's a good sanity check to see, for example, that your data integrated, and that maybe you don't have batch effects that are too bad, but they are really qualitative tools, and so they're not useful for quantitative analysis, which is what you want. And so I say we need tools for visual quantitative data analysis. And so that's what we are going for at WormBase. And these are the four tools that I'm going to show you in the remaining 15 minutes that we are almost ready to deploy. The code is on GitHub. You can try and test it out. We have some test deployments. But it's not in production yet. The preprint is not out, but it's going to be out in like 2 weeks. The first tool is for performing interactive differential expression, and the second tool is for performing gene abundance visualization, which is to say you can see heat maps, histograms of the distribution of the data, and swarm plots. And I'm going to show you what each one of these are. All that I'm showing you now is listed at this address here, wormbase.github.io/single-cell. Please go there, take a look, play around with the tools, and then like give me some feedback if you have opinions, because it will go into the tool. We want this to be useful for people. We don't want it to be just, look, we did a thing, you know? Okay. So the first tool that I'm going to show you is for doing interactive differential expression. And what this means is usually when people do differential expression, you can arbitrarily combine groups of cells. And so it can be hard to, a priori compute all the possible combinations that a user would be interested in. So we developed this tool that I was just doing a deploy on AWS, so it might break, that enables you to do the following. I have here 3 studies from elegans, from single cell, and I told the tool to group them by cell type, by sample description, and by study. And I am able to filter them by which study they came from. And so what this is saying is that the Ben-David 2021 study has 99, distinct sample combinations between like cell type and sample. And then Packer, which is the embryogenesis data set, has 1,000 distinct combinations, and that's what it looks like. So each sample here is one of the batches that they published. This is data from Geo, and then each cell type is according to the author's annotations. And so I'm going to give you an example. Let's say that we want to compare L4 larvae with L2 larvae, which is 2 different stages of development. So Taylor did L4 and Ben-David did L2. And then, so you see here that I only have Taylor here, I only have Ben-David here. And then let's say that I am interested in the AFD neuron. And so I can scroll here and select only the samples that have AFD neuron. And because Taylor did sort for neurons, they have a bunch more batches that have this particular neuron. And so what I'm selecting here is distinct groups from different samples that have a certain number of AFD neurons. And so we show these numbers here so that people can select groups that have at least 50 or so neurons of interest. And so I am now comparing 2 selections of AFD neurons of 2 different life stages. So I probably will not see a lot of difference. I can give my job a name, and I can choose some genes to highlight. So I'm going to choose some neuropeptides to highlight. It will highlight the entire family. And then I submit the job, and what you're going to get back is a gene count matrix. A differentially expressed gene table and interactive plots that you can use to quickly inspect the data. So this is running on an AWS instance that I just deployed. So it has like 8 gigs of RAM. It doesn't have a GPU, which means that you can deploy this locally. You can deploy this on AWS and serve it to people. You can do it just for your lab. The point is that it makes it really easy to offer an interface like this for the biologist in the lab, in the wet lab, that really wants to explore the data to see what's going on. And so we do see that there's like, okay, so this is showing us that there is a bunch of genes that are differentially expressed. And upon mouse over, you were supposed to see the gene name. But obviously, because I did this deploy 15 or 40 minutes ago, I screwed up a little bit. So you do not see the gene name. And then down here, you're supposed to see the gene name for the tables. Okay. Well, just so you know what it's supposed to look like, here's a deploy that I did for another group that I'm helping with data analysis. And I'm just going to select 2 groups at random. And so here you see that like the clusters don't have names because they haven't labeled the cells yet, because it's a different stage of development. And so I just gave them the cluster ID, and the biologists are the one that have the domain knowledge to know how to how it makes sense to annotate the data. And so I just want to show you. Oh, there you go. Okay, so now when I mouse over, it does show the gene description. It does show the gene name, the WormBase ID, and you can download these tables here that have gene name, gene ID. You can sort them by enrichment, or you can sort them by log fold, by p-value. You can sort them by mean expression in the clusters, and then you can download a CSV file. There it goes with your results. Okay, so that's one thing. Oh yeah, and the cool thing is that this tool is, you can deploy it on the command line with like one line. You clone the repo, you pip install from the repo. All that you need is a train the CVI model, and then deploy the model, and you tell it, I want to stratify by study, by sample, by cell type, and it will give you like this kind of view that I showed you. So if your sample contains a different kind of annotation, that's, let's say, life stage, you can also stratify by that. You choose how the options that are going to show up in the data. And just to reiterate, this only takes in a trained CVI model, so it's not a custom data type. It's plug and play with the SCVI framework. Okay, I have 7 minutes. That's plenty. And so I want to show you the other tool that we have been working on. And this one takes in pre-processed data and enables you to visualize the gene expression level of different genes or different cell types. And it has 3 kinds of visualizations: heat maps, gene histograms, and swarm plots that I'll explain separately, because They're kind of like a new thing. That's not a common visualization. So here's the heat map. There's like a example selection of genes. The data that you're seeing here, like it's just test data, so like don't take it super seriously. We're just using it to debug. And the idea is that, you know, when you're comparing your tissues of interest, and then you say, oh, this gene is highly expressed in this tissue, I wonder what it is about. This has a REST API, so it goes and can link directly to the WormBase page. And then there's the gene description, and you can go to other things, or it can link, you know, to the ridgeline visualization of that gene so that you can see its distribution in other cell types. And what you're seeing here is not counts, like transcript counts, but like an inferred expression level that's SCVI infers. So like if you sample 100 transcripts, 1 in every 100 are likely to come from in ADA neurons are likely to come from gene NDO6. We have the heat map style visualization, and we have the dot plot style visualization, which might be a little easier to see than colors, especially if you're colorblind, even with 3Ds. I'm more of a fan of this style of visualization, but it brings up the same kind of data. Then we have the ridgeline plot, which I had mentioned. You can just search for one specific gene that's present in the data, and then it will show you what does the expression of that gene looks like in all the cell types that you have annotated in the data. Oh, look at this. This is probably going to change a little bit the way that we're displaying this data, but the idea here is not to be like super quantitative, but just convey an idea of like, if you have a marker gene that you know should be expressed in one cell type, how does it look in other cell types, right? So you have to have data annotated in a way that you think you trust in order for this visualization to be useful. And then the other kind of visualization that we implemented is called swarm plot. And a swarm plot is a really interesting way to find marker genes in your data. Marker genes are genes that are like highly expressed in only one tissue and are not expressed in other tissues. And the way that this plot works, the data that it's being displayed here is the following. You have a selection of genes here on the left. You show the expression level or the expression rate that is inferred by SCVI, because usually you want marker genes to have high expression. You show the log fold change of that gene in every tissue relative to one reference tissue. So in this case, it's the ADE neuron, and everybody that is to the left of this black line here is less expressed, and everybody that is to the right is higher expression. And so when you mouse over, it will show you the tissue identity, and it will show you like the absolute numbers for the inferred expression and for the log 2 fold change. And so let's show you what it looks like in practice in the visualization. So again, please don't take this seriously because the data that's here is like just a test. So like don't go using it right now to find markers for your cells just yet. This is the data that is brought up on mouse over. And so you see which cell type a dot corresponds to, which gene that in the y-axis it corresponds to, and you see the expression in that cell type, in the reference cell type, in this case ADA, and you see the log fold change in those genes. And so soon we are also going to enable you to sort the genes here by p-value or by expression, and you can already choose certain genes to highlight in this plot. So this is like 90% of the way there. It's missing the last 10%. That's why it's not in production yet. We're still hammering out a few details. Now, why are these swarm plots cool? Because like I mentioned, you can find marker genes. And one thing that happened was I came up with this plot for our lab, for Sternberg lab, to find markers. We selected one candidate from this plot, and for one specific neuron that didn't have good markers, and that was the result. A super bright GFP labeled neuron that got everybody very excited. So that was a very nice result to see coming from public data, the CeNGEN data, that was just out there. If you knew how to process, but also how to visualize the data to make an informed choice about your biological experiment. And then since then, we did a couple more. We did 6 more, and out of those 6 candidates, 4 worked. So it's an incredibly good hit rate. And if you want to use this to find markers on your data, it's on GitHub. It runs on a Colab notebook right here. This example runs with C. elegans data. It explains the stuff that I told you. It will process all the data. It will train the CVI model. It will process the cell types that you want. And then bam, down here, you're going to have your swarm plots. And this ones you can trust. These ones are real. You see that I sorted it by p-value. And so it begins with everybody to the left of the black line. And then as the p-value goes down, they kind of start muddling up. So this you can use. The deployment of the app is still in development. And so with that, I want to say thanks. And again, I want to highlight the fact that the visualization app that I showed you, that work was all driven by Valerio, who's the dev at WormBase. The DE app, it was mostly me, but Valerio gave a ton of input on how to make the code better. And we want your feedback. We really want your feedback. Like, we want to make these tools better and useful for people. And so if you have an opinion about how it could be better, please tell me or email me, or try out the tool and then tell me. But please tell me. That's it. I'm going to stop sharing my screen now.